Good morning, everyone. Thank you for joining GeneTex webinar. This is Liz Hai from GeneTex Taiwan office. Just a reminder for people who just joined us, please silence your microphone and turn off your camera during this webinar. During this event, if you have any questions, please leave your message in the chat box at the bottom of the right panel. We will have a Q&A section at the end of this slide and address these questions. So today's topic is enhancing anti-cancer immune response by targeting glycosylated PD-L1. PD-L1 is a key molecule that involves in the immune checkpoint pathway, and therapies have been developed targeting this protein for cancer treatment. We are happy to have this opportunity to invite Dr. Jiawei Li to give his talk and share his knowledge about PD-1 pathway and breast cancer.
Hi, good morning, everyone. Thank you so much for giving me this great opportunity to present some of our recent study on the PD-L1. The field of immuno-oncology has drastically reshaped the landscape of cancer therapy since the development of antibody blocking immunocheckpoint protein, which attenuate anti-tumor immunity by inhibition of T-cell activation. To date, antibody targeting immune checkpoint protein such as PD-1 and the PD-L1 have been approved by FDA to treat over 25 types of cancers and in over 200,000 patients. Despite their clinical success, issues such as missing targets, intrinsic and acquired resistance, hyperprogressive disease, and the lack of reliable biomarker or autoimmune disease has now become a new challenge await for further investigation. So let me use this slide briefly show you the concept of the cancer immunotherapy, which is developed by the Dr. Jim Allison and Dr. Hundra, and who actually won the Nobel Prize in the 2018 for their innovative identification using monoclonal antibody targeting the inhibitory signaling pathway in the tumor microenvironment in the case of PD-L1, and thereby enhance the anti-tumor immunity as a new therapeutic agent. So in the tumor microenvironment, we can see that the cancer cell, when you engage with the T cell, the T cell will surveillance in the tumor microenvironment, try to recognize the new antigen on the T cell and try to remove this foreign substance. So cancer cell, on the other hand, is very smart. They try to escape this immunosurveillance by inducing the PD-L1 expression on the cell surface. And this PD-L1 will engage with PD-1 receptor on the T cell and thereby suppress the T cell activity. Scientists use monoclonal antibody to block PD-L1 or PD-1 to disengage this interaction and thereby activate the T cell in the tumor microenvironment and then allow the T cell secrete anti-tumor substance such as granzyme or interferon gamma to further fight against to eliminate the cancer. And this process called anti-cancer immunity. And this approach has actually been very successful. So pardon me for showing this not outdated list, but until 2019, there are already over 5 monoclonal antibody actually targeting PD-1 or PD-L1 that have been developed by many pharmaceutical companies that also be approved by FDA to treat several types of disease such as myeloma, lung cancer, or even which is known to have the provide immune heart environment for the cancer immunity. So in this slide, it is important that for each antibody that actually they pair with anti-PD-L1 assay antibody. For those patients who use this biomarker, shows a biomarker positive, they are suitable for certain type of antibody treatment. And most importantly, FDA actually approved the treatment of using atezolizumab in triple negative breast cancer cells combined with the nab-paclitaxel in the 2019, which is related to the today's topic. So that's why I specifically point out. So regard the clinical success, however, the general response rate for the anti-PD-1 or PD-L1 is about 20 to 40%, depends on the type of disease we are dealing with. So means there is still tremendous need in understand the underlying mechanism, identify the good biomarker, and also find a good combinational therapy to try to enhance the response rate of the immunotherapy. So the type of cancer we are of interest is breast cancer. This is actually the most devastating disease in the woman. And as this slide show in the American Cancer Society year 2020, in the female, actually you can see most patients in the female aggressive type of cancer diagnosis, breast cancer is listed as a #1 identification of the new cases. In terms of the mortality, the breast cancer is still among the #2 top killing, the damage to the devastated disease in the older woman. And of course, in the past 20 years, the treatment of breast cancer has been significantly improved by many scientists that develop a new therapeutic agent. And the 5-year survival rate is over 90%. But those patients, now they still start to live longer, but they still suffer from metastasis or relapse from the treatment. And it is known that on average, 1 out of 4 women in their lifetime, they will face some kind of breast cancer. So therefore, this disease is still very devastating and need our attention to further investigate. So in general, breast cancer can be divided into 4 subtypes. They are ER positive, PR positive, HER2 positive, and one type of breast cancer that lack of any these tumor antigen called the triple negative breast cancer cells. In the past 20 years, physician scientists has been put a lot of effort to try to identify new therapeutic agent to treat ER positive and PR positive and the HER2. And so for a clinical, right now the physician choose the tamoxifen to treat the ER positive patient, HER2 antibody suitable for the HER2 positive. However, this triple negative breast cancer, which lack of any tumor antigen, so it's become a new challenge. It's very difficult to treat it. And the standard therapy for this triple negative right now is rely on the chemotherapy. And only half of the patient, triple negative breast cancer patient, has made response to the chemotherapy. So therefore, there is important, and also there is 15% of the patient actually diagnosed with triple negative. So therefore, there is tremendous need in understand the molecular mechanism of triple negative breast cancer. So one thing out of our interest is that when we compare the tumor microenvironment across this breast cancer subtype, triple negative breast cancer has the most infiltrated T cell compared to other subtype. However, regardless, there is a most expression of the T cell in the tumor microenvironment. This type of cancer still progress, suggesting there must be a strong immunosuppression mechanism in the triple negative breast cancer cell that try to promote the disease progression. So therefore, it is an interesting subtype of breast cancer that is suitable for study the cancer immunosuppression response. So in the 2014, we are trying to understand the underlying mechanism of the triple negative immunosuppression. So the one molecule across our mind is PD-L1. So in that time, we try to first study all the literature report that PD-L1 is expressed at the 33 kilodalton molecular weight, which is relevant to its nucleotide that encode from 290 nucleotide that encode for 33 kilodalton of the PD-L1. So the first experiment we do is we take several cell lines and run a quick Western blot. So in this original slide, we actually can see it's very difficult to see PD-L1 expression at the 33 kilodalton. And also there, but interesting, there is a huge protein expressed at the 50 kilodalton molecule out in the Western blot. So in that time, in order to really detect the PD-L1, we always have to cut off the membrane, try to discard the upper 50 kilodalton non-specific pickup, and in order to enrich the expression signal of the PD-L1 at the 33 kilodalton. So then we move to another method to investigate the PD-L1. That is just clone this 290 nucleotide 33 kilodalton DNA, cDNA, and the transient transfer in the 293 cells, and then do the Western blot. And to our surprise, this PD-L1 does not express at the 33 kilodalton. Instead, this overexpressed PD-L1 also expressed at the 50 kilodalton molecular weight. So therefore, we start to speculate maybe the 50 kilodalton molecular weight on the Western blot is the true PD-L1 expression signal. To further validate this hypothesis, we use shRNA and the CRISPR-Cas9 knockout PD-L1. When we knock out this 33 kilodalton of the protein, we actually see the PD-L1 at the 50 kilodalton molecular weight disappear, which suggests that maybe the 50 kilodalton molecular weight of the protein is actually PD-L1. Then we look into across a different cell type of the breast cancer and investigate this 50 kilodalton molecular weight of the PD-L1 signal. We found that in the triple negative breast cancer cell, most of the PD-L1 is heavily enriched in the expressed at the 50 kilodalton molecular weight. In vice versa, non-triple negative breast cancer actually has more 33 kilodalton and slightly 50 kilodalton of the PD-L1. And we also, it is known that the triple negative breast cancer has high EGFR expression. We also look at the EGFR expression level and see there is a correlation between, across the different cell line, between EGFR and the 50 kilodalton molecular weight of the PD-L1 expression. So then we further look into the literature to find out why cause the PD-L1 goes to 50 kilodalton molecular weight. And turns out PD-L1 is a membrane protein. And a lot of mammal protein, particularly membrane protein, actually is heavily glycosylated. So therefore, we take the one unlinked glycosylation inhibitor called tunicamycin to apply this drug in our overexpression system. As you can see in this slide, when we increase the concentration of the tunicamycin in the 293 cells, which express 3 different types of PD-L1 that contain different tags, we consistently see in the presence of tunicamycin, PD-L1 now goes from 50 kilodalton molecular weight down to 33 kilodalton molecular weight, suggesting PD-L1 is heavily glycosylated in the mammal, in the cells. So why is protein glycosylation? It is a kind of attachment of sugar moiety to protein and thereby form a glycoprotein. as I mentioned, 1/3 of the mammal protein actually is highly heavily glycosylated. This unlinked glycosylation glycosylation event is predominantly occur at the NXT motif on the protein, where the N means is asparagine, X is any amino acid, T and the S is serine and threonine. And glycosylation is important for biosynthetic secretory pathway, as we show in the right cartoon, that when the protein is synthesized, during the protein synthesis, the polypeptide in the ER will be attached with a type of glycan on the surface, and therefore lead the protein secrete from the ER and go to the Golgi toward its destination on the cell surface. Especially in this case is PD-L1. And it is known glycosylation is important for protein folding, protein localization, protein-protein interaction, and some of the glycosylation actually control the enzyme activity of the protein. And correlate to the recent study of COVID-19 pandemic, glycosylation is also known to be important for certain type of virus infection, such as alpha-2,6-sialidation is important for the influenza virus enter to the cells. So therefore, there is a tremendous need in understand the very complex structure of the protein glycosylation. So to further validate whether PD-L1 is glycosylated, we actually check the PD-L1 sequence and found out in the extracellular domain of the protein contains 4 NXT motifs. They are N35, N192, N200, and the N219. When we then create the site-derived mutagenesis, try to mutate each asparagine into glutamic acid. When we mutate individual single glycosylation site, we can see the PD-L1 expression molecular weight start to reduce. We create double mutant and triple mutant till we complete replace all the 4 asparagine motif on the protein. We now start to see PD-L1 can go to 50 kilodalton molecular weight, which runs along with the signal that from wild-type PD-L1 that treated with tunicamycin. We also create another 3 non-NXT motif asparagine mutation, and they do not change the PD-L1 expression at 50 kilodalton molecular weight. So these results suggest that PD-L1 is heavily glycosylated at 4 distinct asparagine motif. So what's the protein glycosylation due to PD-L1? So one thing out of us for the biochemical analysis, we actually want to understand whether this glycosylation changes the protein stability. So we do a simple experiment by using the cyclohexamide to do the post-stress experiment to see to detect the protein stability of the PD-L1. In 2 different cases, in the presence of tunicamycin, we can detect both glycosylated and non-glycosylated PD-L1. And in this chart, we actually can see non-glycosylated PD-L1 is undergo fast protein degradation. We also did a ubiquitination assay by pulling down the PD-L1 from the cells in the presence of NG132 and tunicamycin. We can see non-glycosylated PD-L1 actually is heavily enriched with polyubiquitination chain. These results suggest that non-glycosylation of the PD-L1 actually is undergo fast proteasome-mediated degradation, which make a lot of sense to our earlier study in the cell that we barely see the non-glycosylated protein, whereas in the many cell line, cancer cell line particularly, PD-L1 is heavily enriched at a 50 kilodalton glycosylated molecular weight. Then we look into the further analysis underlying mechanism. So let me just summarize our finding with this slide. We actually found out in the extracellular domain of the PD-L1, there is a very consensus GSK3 beta binding motif right adjacent to the 3-NXT glycan moiety, which is very evolutionary conserved. GSK3 beta binding site together with a beta TRCP binding motif. And we did quite a lot of analysis. So let me show you in this model, that is, we hypothesized that PD-L1, non-glycosylated PD-L1 actually will be recognized by GSK3 beta mediated phosphorylation. This phosphorylation will further recruit the beta TRCP, come close to the PD-L1, forms a induce the polyubiquitination of PD-L1, pump PD-L1 into the cytoplasm, and thereby undergo fast proteasome degradation. When PD-L1 start to glycosylate, this glycan structure on the PD-L1 may create a steric hindrance to GSK3 beta, and thereby keep GSK3 beta away from PD-L1, and then allow the PD-L1 to be stabilized and then go to the cell surface to interact with PD-L1. So we have, in the past 5 years, at the MD Anderson Cancer Center, we actually done quite a lot analysis focused on the post-translational modification of PD-L1. And we actually found out there are 2 enzymes that are important for the glycosylation, B3GNT3 and the STT3A. These 2 enzymes will upregulate PD-L1, then allow the stabilize the protein and have contribute to the immunosuppression activity. We also identified that GSK3 beta, AMPK, and the JAK1 also specifically phosphorylate on the PD-L1. In terms of the polyubiquitination, we identified beta TRCP and the SPOP as an enzyme. We also found out the ubiquitinase enzyme such as CS5 and CNTN6, as well as the palmitation enzyme. So those actually play important role in regulate PD-L1 and contribute somewhat in the PD-L1 for their protein stability, and thereby provide a strong rationale to design combinational therapy. So we have published this paper, this review in the Molecular Cell in the 2019. So this is a slide summarize all the current knowledge about the regulatory mechanism of PD-L1 through transcriptional regulation and the post-translational modification. So the next we would like to know is what's the function of glycosylated PD-L1. So it is known that the PD-L1 is important to interact with PD-1 on the cell surface. So the first experiment we do is we just do the co-immunoprecipitation by putting PD-L1 and PD-1 together in the tube, and then to pour down the glycosylated or non-glycosylated PD-L1. What we found is that only the glycosylated PD-L1 has a strong interaction with PD-1, whereas PD-L1 lack of glycan structure, they do not interact with PD-1. We also did a T cell killing assay by using the B16 overexpressed with OVA peptide and also the nuclear RIP signal and then we also, on the other hand, we purified the mouse T cell from the OT1 mice to provide the antigen specific killing effect. So in the panel B, we can see that the cancer cell express glycosylated PD-L1 actually have a very strong suppression activity toward the T cell. So in this case, the green color, green fluorescence color represent the caspase-3 activity, which is recapitulated cell death activity. When the cancer cell expresses non-glycosylated PD-L1, it now becomes very sensitive to T cell killing. So this result actually suggests glycosylation has an important function to PD-L1 mediated T cell suppression. So the next question is it possible to develop a therapeutic strategy to target this glycosylated PD-L1? So we actually work with the SDQ pharmaceutical company to isolate the antibody that specifically recognize glycosylated form. So in this case, we inject the biopsy mice with glycosylated PD-L1 as an antigen to immunize the mice to produce hybridoma, And the first experiment we do is we use ELISA to screen the hybridoma clone that only specifically recognize glycosylated PD-L1, but not those non-glycosylated PD-L1. Then the next experiment is we develop a in vitro using the time-lapse microscope to detect the PD-1 PD-L1 interaction, and they'll use this antibody to neutralize PD-1 PD-L1 interaction. So in this case, under time-lapse microscope, in the 1 to 12 during 12 hour period of time, we actually can see the PD-L1 and the PD-1 interaction under time-lapse microscope with this signal. In the presence of anti-glycosylated PD-L1 antibody, this interaction will be neutralized. So in this representative, it shows 4 antibody clone that is able to specifically neutralize PD-L1 and the PD-1 interaction. So the next, we want to isolate this antibody that can specifically internalize into the cells. So in this experiment, we actually label the antibody with a pH rhodol, which is a fluorescence dye. When the antibody internalizes into the lysosome, the pH value change the acidic condition of the lysosome, actually allow the pH, the linker to be cleaved, and thereby induce the red fluorescence in the cell. So in this case, we can use a time-lapse microscope to detect the red fluorescence by indicating this antibody actually has a strong internalization ability. So let me show you this slide that is there are several clones. So we are specifically interested in the STM108 and the STM073, these 2 clones. We show these 2 clones has a very strong neutralization effect by reducing the PD-L1 and the PD-1 interaction. We also create a subset of the protein that specifically express one glycan structure on the PD-L1, but not the other 3. In this subsequent clone, we actually can see that the our STM108 antibody has a specificity to interact with N192 and the N200 glycan structure. And when we compare with our earlier mass spectrum analysis data for the PD-L1 glycan structure, we actually found that the N192 and the N200 glycan has a very unique motif, glycan motif that is polylagnate. And as I mentioned earlier, these 2 antibody clones has a very strong internalization ability. And we also investigate whether the glycosylated PD-L1 is more specific to immunosuppressive the cell. So by doing this way, it is known that PD-L1 also is expressed in the normal cell. We actually hypothesize glycosylation is heavily appeared in the negative, the inhibitory signal, immunosuppressive cells triggered by EGF. So therefore, we found our STM108 antibody has a specificity to those immunosuppressive phenotype to detect this immunosuppressive phenotype. So in the end, we would like to take advantage of recent developed strategy called antibody drug conjugate. So in this case, we have our own anti-PD-L1, glycosylated PD-L1 antibody. We choose NNAE, which is a cytotoxin that is similar to that is a mimic to the anti-tubule drug. On average, we conjugate 4 NNAE molecule on the glycosylated PD-L1, and then we test the binding specificity of this antibody using the in vitro cell line system. We found out only the cell express glycosylated PD-L1 that has a very strong toxicity by the new antibody drug conjugate. We also use the animal model that is 41 cells overexpressing human PD-L1, either glycosylated form or non-glycosylated form. We found that this antibody can specifically eliminate cancer cells that express glycosylated PD-L1. And more importantly, in this mice survival data, we can see the naked antibody, which is glycosylated PD-L1, can increase mice survival rate about 30%. In the presence of antibody drug conjugate, we can further increase the survival of the anti-tumor immunity, increase the mice survival to 80%. That is very a significant improvement. We also did the IHC analysis to look at the apoptosis, and as well as the cytoph analysis to specifically find out our antibody PD-L1 drug conjugate actually can specifically eliminate cancer cell that has a very low toxicity to the normal cell, such as immune cell. So in the end, we would like to use this cartoon to summarize our study. That is, in the triple-negative breast cancer cells, the high EGFR signaling may induce a subset of the glycotransferase. In this case, we know it's B3GNT3 that can catalyze polylagnate glycosylation on the PD-L1. Allow the PD-L1 go to the cell surface to interact with PD-1 to suppress T cell immunity in the triple-negative breast cancer microenvironment. If we develop this anti-glycosylated PD-L1 with antibody drug conjugate, we are able to exert at least 3 activities. One, this antibody can block PD-L1 and the PD-1 interaction, allow the T cell to reactivate, and then fight against the cancer cell. The second activity comes to the antibody that can cross-link the cell surface PD-L1 to an extent allow the PD-L1 translocate, internalize to the lysosome. And this internalization will further cleave, activate this product of NNAE, and therefore induce a cytotoxin effect to the triple-negative breast cancer cells. And most importantly, this cytotoxin NNAE is known to induce bystander effect. This cytotoxin may release in the tumor microenvironment and further eradicate those cancer cells that do not express PD-L1. It is known that PD-L1, only 20% of the cancer cells express PD-L1. So therefore, using this antibody drug conjugate actually has a very wide application to completely eliminate the cancer cell of the triple-negative breast cancer cells. And we have recently licensed this product to from ST with our work with ST Cube. Actually is has been recently licensed to Top Immunity and Alps Biotech to further develop our antibody into clinical trial, into clinical phase. So another important question is it is known that there are many immunomodulators, such as PD-1, PD-L1, and they are also co-activatory signal. So this slide briefly show in the field there are many immune checkpoint has been identified. So since our development in the glycosylation of PD-L1, we would like to know if other immune receptor also share a similar glycosylation pattern. So the first experiment we do is we take several important immune checkpoint, and we use PNGSF, which is an enzyme that can specifically remove the glycan structure. And then we run the Western blot to see does the molecular weight of those protein purified from the mammal actually can has a molecular weight reduction. And the answer is they are all heavily glycosylated. And we then set out an experiment to test their interaction by receptor and covalent ligand. And we remove the glycan structure and then test if these receptors still interact. So to our surprise that in the case of inhibitory signal, removed glycosylation block their interaction. And whereas in the case of activatory signal, if we remove the glycosylation, it does not seem to affect co-activatory signal. So these results suggest maybe targeting glycosylation, inhibition glycosylation can reactivate inhibitory signal, but not affect the existing co-activatory signals. So in the end, our study actually pinpoint that PD-L1 is heavily glycosylated in the cancer cell. And glycosylation of PD-L1 can provide stability to the protein, allow the PD-L1 go to the cell surface, and thereby suppress the T cell function. If we can develop an antibody that specifically recognizes PD-L1, we are able to cross-link the cell surface PD-L1 molecule and therefore induce the protein internalization to the cancer cell. By using antibody drug conjugate, we are able to deliver specific cytotoxic effect to those heavily glycosylated PD-L1, particularly cancer cell, their triple-negative breast cancer cell, to eliminate these cells with strong bystander effect. So lastly, I would like to take this opportunity to thank a lot of people that have been contribute to this project while I was at the MD Anderson Cancer Center in Dr. Ming-Chi Hang's lab. And now my lab is at the IBNS at the Academia Sinica in Taiwan. So we are continually trying to understand, investigate the underlying mechanism of glycosylation in PD-L1, or in the recent study focused on COVID-19, such as ACE2 and the spike protein glycosylation. We think there is a strong application, there is a great potential. This glycosylation has been ignored in the past, so we would like to put our effort to investigate and further increase the response rate of PD-L1, or to seek for novel therapeutic methods maybe targeted to help against the COVID-19 pandemic. So thank you so much for your attention. And if you have questions, please feel free to ask. Thank you.
Thank you, Dr. Lee, for giving this informative talk. So before we enter in the Q&A section, I would like to take a few minutes to introduce GeneTex and reagents we developed for life science research. So in 1997, GeneTex was founded in Texas by 3 scientists. And at that time, the completion of Human Genome Project has fastened the development of life science. So 10 years later, the company added a new facility in Taiwan to further expand production capacity to meet the increasing needs. So at the same time that we had changes in the US as well, the office was relocated to Irvine, California, which is closer to the San Diego Life Science Cluster, close to more institutes, research institute, and also biotech companies. And gradually, GeneTex has become an internationally trusted antibody manufacturer. So the GeneTex catalog started with antibodies for DNA repair and breast cancer research, which is related to the talk today. And GeneTex keeps expanding its product coverage, and now we offer quality antibodies for other research areas, including cell death, epigenetics, neuroscience, and infectious disease, and also including antibodies for SARS-CoV-2, the virus that caused the current pandemic. And around 50,000 products in total. The majority products are primary antibodies. And for many of the antibodies, a small size 25 microliter is available for trial. Serving the research community, GeneTex takes product quality seriously and has implemented 5 methods to validate our products. So the first is knockdown validation. It is a very straightforward to see that as a signal is detected on the wild type and lose the signal in knocked out samples. So this method is well received by researchers and customers. Secondly, comparative antibody is another approach that we use. Comparable antibody basically is using 2 or more antibodies against the same target, usually against different epitopes of the target. So when the results are consistent, it is convinced that the antibodies are giving the right signal. And the third one, IP-mass, is to use an antibody in an immunoprecipitation assay and analyze the protein captured by the mass spectrometry. And this validation is done through a collaboration with a professor in Norway, and the results are also published. So the next biological and orthogonal characteristic method, it is to test the antibody using treatments based on biological characteristics that are already known to the target protein. So like by using drug treatments, subcellular fractionations, etc. And the last one is protein overexpression test. So testing the antibody on target protein overexpressed lysate. So at GeneTex, this method is mainly used to confirm cross-reactivity of the antibody. So we would also confirm the antibody can also detect endogenous protein expression level at the same time before it can be released. So the 5 icons here are used to label the antibodies in our catalog and website. So easier for customers to identify what method of the protein, the antibody has been tested with. And here I would like to introduce some frequently used tools and antibodies in cancer immune checkpoints and also breast tumor marker studies. So let's start with PD-L1 antibody, the main focus of today. So this antibody, GTX-104763, is a rapid polyclonal antibody. So as what Dr. Lee just presented, PD-L1 is expressed in cell and glycosylated. Glycosylation is important for PD-L1 and PD-1 interaction. This antibody can detect the glycosylated form of PD-L1 around 50 kilodalton, and after tunicamycin treatment, it can detect the glycosylated PD-L1 at around 33 or 34 kilodalton. So further, we tested the antibody using siRNA knockdown test as performed to confirm the specificity of the antibody. So, and through this overexpression test on PD-L1 and PD-L2, which is another ligand of PD-1, the result shows that this antibody only recognizes PD-L1 and not PD-L2. So it is also known that PD-L1 is highly expressed in some cell lines, like the triple negative breast cancer cell line here, MB231. So we also use different expression level cell lines in the same plot to confirm whether the signal detected is correlated to this non-biological characteristic. And further, by a side-by-side test with an antibody from CST, which is a well-cited antibody, the GeneTex antibody is presenting a robust signal in a 1 to 1,200 dilution, and comparing to the CST product, it was diluted 1 to 500. In addition to Western blot, this antibody is also validated for cell staining and tissue staining for both frozen and paraffin fixed tissues. So here is the IgCP staining, which shows a nice membrane staining pattern of the tissue tested using human ovarian carcinoma. So comparing to the CST product here, GeneTex product got a cleaner background and a sharper signal with a 1 to 1,000 dilution, while the CST product was used at 1 to 50 dilution. So GeneTex also offers golden marker for other immune checkpoint proteins, such as PD-1, the receptor of PD-L1. So this antibody has been validated on IgCP and Western blot for human and mouse tissues. The IgCP here shows membrane staining pattern of inflammatory cell in human lung cancer. CCL4 appears to act as a negative regulator of T cell activation, and it is another target for cancer immunotherapy. So for this clone, BNI3, we offer various fluorescence conjugation formats for flow cytometry purpose. And this CD4 antibody, it works in neutralization assays and can detect CD4 positive T cell activation, and has a good performance in both IHC and flow cytometry. And next, let's move on to IDO1. High IDO1 expression positively correlates with microvessel density and poor prognosis in breast cancer patients. So after an inducer interferon gamma here treatment, we can see the signal enhanced in the Western blot results, and the GeneTex antibody can detect a stronger signal at the dilution 1 to 3,000 dilution, comparing to the CST antibody, which was diluted 1 to 50. So the next section will be about tumor markers for breast cancer. So including HER2, estrogen receptor, and progesterone receptor. So these three are markers used for breast cancer subtyping. So first of all, HER2, which also known as NUT or RBB2, it is a member of EGFR family of tyrosine kinase. So GeneTex antibody GTX 10509 is specifically detects HER2 protein, and it is confirmed by a cross-reactivity test using overexpression samples. And the results demonstrate the antibody does not cross-react to EGFR, which shares a very high identity with HER2. And also the specificity was also confirmed by using knockout lysate. Also, the antibody detects a nice cell membrane pattern in HER2 positive breast tumor cancer tissue sections. And this side-by-side comparison between this antibody and the CST product was performed at 1 to 500, and the other one is to 1 to 100 dilution, respectively. Estrogen receptor alpha and beta, they have been extensively studied and targeted for the development of therapeutics against hormone-positive breast cancers. So GeneTex estrogen receptor alpha antibody 1F3, it is a mouse monoclonal antibody, which was the sensitivity and specificity has been stringently validated at GeneTex and also has been used in multiple publications. So the estrogen receptor beta antibody 14C8, it is one of the most commonly used reagents to detect this target. So accumulate, we had more than 100 citations for this clone. The two antibodies, they are the specificity has been tested and they are specific to its target and do not cross-react to each other. So it's confirmed by overexpressed lysate of ER beta and ER alpha. So both of them, they are suitable for Western blot, immunohistochemistry test, staining, and also flow cytometry. And on top of that, the ER beta antibody can be applied in chip assay, and it is the most cited ER beta antibody on CyDAP. So next, the progesterone receptor, PR, it is a well-known estrogen receptor target gene. So it's expressed in over 2/3 of ER-positive breast cancers. So this GeneTex antibody, it was developed for immunocytostaining and also tissue staining. So the result shows that it distinct staining on PR negative and PR positive breast tumor tissue sections here. So it is the signal was only detected in the PR positive breast cancer. Last but not least, the EGFR is another common breast tumor marker. EGFR is linked to poor outcome in some breast cancer cases. So here we show a side-by-side data plotted with the monoclonal antibody GTX-62887 and got a cleaner signal than the CST antibody when plotted diluted at 1 to 5,000. So the sensitivity and specificity has been stringently validated by knockdown and also cross-reactivity test here. So also here we got an IGCP result and tested on human ovarian carcinoma. So that's some highlight products for immune checkpoints and breast cancer research. If you are interested in any further information, you can either contact GeneTex or our local distributors here.